probably invite you to turn on your cameras after the seminar for the Q&A session so we can see you. So now we will begin. So good morning, good afternoon to everyone joining us here in person and online. Welcome to this special edition of the Alliance Science Seminar. We are especially pleased to have colleagues from both inside and outside the Alliance with us today. Thank you for being here. This session is being held in honor of the International Day of Banana, which officially took place in April 16th, but as you know, there are a lot of festivities going on. So we moved the celebration to today, April 23rd. Our topic today is leveraging genomic tools to harness banana diversity from Genebanks. We'll be hearing from Mathieu Rouard over there. He's a senior scientist in genomics, computational biology, and data management, who leads the genetic resources characterization team within the Banana program. He'll be sharing insights on how advanced genomic approaches are used to characterize and document banana gene bank materials, strategies that not only enhance conservation, but also promote the practical use of banana diversity. A quick note before we begin, today's seminar is hybrid, as you may notice, and we'll take questions from both our in-person and online audience right after the 30-minute presentation. We also have simultaneous interpretation available, so please feel free to activate the feature if needed. We have simultaneous interpretation online, but also in person here. And now to introduce today's speaker and today's topic in more detail, I'd like to hand it over to Sebastian Carpenter that is connected online. Over to you, Sebastian.
Thank you very much. It's a great pleasure to celebrate this International Banana Day with all of you together. It's nice to see that there is also many interests from outside our organization. So I will not take a lot of your time, just briefly introduce our International Banana Research Program. As you can see from these slides, we are a bit spread over all the world. So we have several offices. We have, of course, our gene bank, but we are really proud that we are have the honor to manage the biggest banana gene bank in the world. And our aim is really to get more banana diversity into our fields. And of course, before we can do that, we need to study, we need to evaluate this diversity. So we need to understand their useful traits. And so I invite you to do this together. And of course, across the world, we are doing this together mainly with national agricultural research partners. So as you see, we have a big office in Belgium around the gene bank. We have a big office in Montpellier where Mathieu is based and who are developing all the tools to genetically characterize and also to manage also the information and to share the information with the world. And of course, as you know, banana is also a very important staple crop in Africa. So we mainly focus on Central and East Africa, where we also have, together with the National Agricultural Institute, NARO, a field gene bank of more than 400 different, mainly focusing on the East African highland bananas. So that's a very important point as well. And of course, our office in Siat and Cali is very important as well, and where Mathieu is for the moment hosted. And so yeah, if I can go to the next slide. So the teams are separated, of course, around the conservation of and the management of the genetic conservation. Of course, we have as Mathieu will explain, the genetic characterization of everything. Very important. We have the evaluation of the different traits, both for climate change and for diseases. And of course, we have all the new technologies around new management techniques and recognizing and manages diseases. So everything around more shift towards agroecological practices, different management techniques, and of course, managing the diseases recognizing and manages the diseases that are all over the world. So centrally on this slide, you see basically, of course, our gene bank. That's the biodiversity that is there, that we are managing, that we are studying. And I'm inviting all of us to study that together. And of course, our knowledge on the diseases and on the agroecological management systems that are available. And then, of course, you see all research around that in the end basically ends up with farmers. It's very important if we want to get this diversity into the fields. We need to work together with farmers and with NGOs and with national agricultural research institutions. And so the difference, my dream is basically that we have in the different agroecological systems, the different unique packages of banana diversity that work locally towards market demands, towards nutrition value, towards also disease resistance, and not every country struggles with the same diseases. And of course, towards the climate, especially water, is also a very important point. So we are trying not only to understand this diversity and where it can be useful, we are also trying to improve it, of course, in collaboration with breeding programs from colleagues, but of course also through modern breeding techniques as gene editing. And of course, we do realize that our gene bank is not complete. There is still very interesting banana resources out there that we do not have yet. And of course, we are continuously, together with partners, looking into how to improve that and how to still fill the gaps in our gene bank. As Mathieu will explain, it's super important to also have, from the genetic point of view, a backup to help us into making decisions, because it's quite it demands quite some resources to start studying also, so we need to prioritize. And so Mathieu will focus in his research into how the genetic characterization makes us much wiser into what are we managing, so how big is the diversity. And of course, if you combine that those genetic markers, let's say, and those insights about the ancestors towards traits, then you get into hands very powerful tools to study things together and to discover things. I will not take longer of the time. Please, Mathieu, the floor is yours. Thank you.
Yeah, so I was saying that I was very happy to be here in person for this Internal Banana Day, and that the main idea of the seminar is to convince you, if you need to be convinced, that banana is a fascinating crop. And just to remind that banana, as you must know, has a beautiful morphological diversity that you can see all over the place. It has a lot of interesting biological features. It's vegetatively propagated. Edible bananas are parthenocarpic, are low fertile. There are different levels of ploidies. It can be diploid, triploid, tetraploids. And you can do many things with banana. You can use the fruits, of course, as raw. You can cook it, but you can use the leaf. You can make bread. You can make beer. You can make clothes. You can make plenty of things with banana. And that's very a fascinating crop for that. It's also associated with a long history in Central America, particularly. So, and I invite you to look at this book from Dan Koppel, which is very interesting. So I recommend it if you want to know more about the background of bananas. But for sure, bananas are a key crop for food security. So it originates from from Southeast Asia and Oceania. It's a very important staple crop for Africa, with plantain, with East Island African banana, with mshare. And it's also very important as an economic crop for Latin America. So being here in Colombia, we'll focus on some traits in Latin America, starting by Fusarium wilt. That is a very devastating disease that has migrated from the east, from Taiwan to the east, and that unfortunately reached Colombia in 2019. It was then discovered in Peru and now in Venezuela, so it's a very important disease. Now here, it stays in the soil for decades. Jorge could tell you much more about that with the Alerta project. So it's an important trait for this continent, and that's something that we need to address and to find solutions. Also, this article that is very recent, published in 2025 in Nature Food, about prediction of climatic changes. So looking at the increase of temperature, looking at the soil, looking at the existing demographic population in Latin America, they could predict that there will be a lot of area where we will have a loss of suitability for the cultivation of banana. So this is going to be a big concern as well. You have some predictions that you can see on the left here, where you can see some prediction on yield. And unfortunately, the prediction for Colombia are not very good in terms of yield for banana in the country. So here again, that's something that we need to take into account for the challenges that are ahead of us. Then another trait is also the monoculture. So the industry in Latin America is relying mostly on one cultivar group, the Cavendish. So it makes it very vulnerable to disease. There is a need for a lot of fertilizer. So very narrow genetic diversity. And however, in Latin America and in Colombia especially, we have a lot of diversity. And that's something that was demonstrated during the last COP in November, organized here as a side event by Jorge and Lorena and Sebastian, where we could see the diversity. And I'm sure we will see again today on the campus a lot of diversity. And we believe that diversity is important. We believe that combined with innovation, it can bring very important solutions for those challenges. So this diversity exists all over the world, in the tropics and the subtropics in germplasm collection. If we focus in Colombia, we have next door the Agrosavia, where we have a beautiful collection. We have Fede Platano, which is not very far from here. So each of those collections have different size, different diversity. And at the Alliance, we are very fortunate to manage the International Transit Center, where we have the largest collection of banana, the largest diversity. So in terms of composition, we have more than 1,700 accessions. So most of them are long races, cultivated banana, 75%. And we have about 16% of wide gene pools and a bit less than 10% of breeding material. So this number evolving because we also organize some collecting mission on farm studies as well. So, but our objective in the team is really to characterize this diversity and to promote the use of this diversity. And to do that, it's important to understand the domestication and the diversification of bananas. So the usual diagram for domestication is this one. So we start with some white species that are diploid species, Musa acuminata for A, Musa balisana for B, Musa schizocarpa for S, and Australimusa textilis for T. So there was a number of introgression, interspecific and intraspecific crosses that led to other diploids that are edible diploids that became parthenocarpic with some vegetative propagation. So AA is when only subspecies of Musa acuminata cross together. AB is when you got some interspecific crosses between A and B, etc., etc. And then, after a number of events, unreduced gametes and so on, it led to the creation to the triploid, where you have most of the cultivated banana that are denoted by AAA, AAB, or IBB. And then some diversification of them. So, and some years ago, we were very much interested to better understand the transition between the wide and the diploid edible bananas. So my colleague Julie Sardos conducted this study that was published and that looked at the history of hybridation in cultivated edible banana from Musa, originating from Musa acuminata, the AA bananas. And what we could see that there was a very important center of diversity in Oceania. So it shows the importance in domestication of Papua New Guinea, because many diploid banana have a background with Musa banksii. We could see also the spread of introgression, and we could demonstrate that there was a lot of introgression, much more than we expected. And we could also confirm that there were some unknown gene pools that are like undetermined here. Those gene pools were unknown in the contribution of banana, confirming confirming some report from our colleagues from CIRAD 1 or 2 years before. And talking about our partners at CIRAD, at this moment, at that period, they started to develop a method that we call in silico chromosome painting. And I will explain basically the principle is that, and Guillaume Martin was the main person working on that, is that for representative of the ancestral gene pools, it was possible to develop SNP diagnostics and to give them a color based on their origin. So it was the creation of this data set. And then a software was developed to look at this SNP in the cultivated bananas along the chromosome. What you see here is a chromosome. And for each SNP to calculate the SNP ratio and to provide a color based on the origin of the ancestral gene pool, right? So combining both the color and combining the SNP ratio, according to the ploidy, because we have multiple levels of ploidy, it was possible to create this type of karyotypes that we call mosaics, where you can paint along the chromosome based on the origin of the ancestral gene pools. So those Musa acuminata, balbisiana, the different subspecies, and so on. So this work was very important, gave a lot of insight for domestication and also the molecular basis for recombination in bananas. But we wanted to go a bit further and to apply that to the international collection. So we screened a big sample in the collection, trying to get most of the diversity. And we could screen most of the groups that you can see here on the PCA, looking at the proximity between the different groups. But for each of the groups, we could develop a particular card with a passport data that was very well curated with pictures, and also this in silico chromosome painting of the group. So doing that for all the different groups, we could combine that and make a catalog that is available on CGSpace, which is now a baseline for pattern for identification in the collection. So each time we have new accessions that we genotype, we produce a SNP marker, we can conduct this type of analysis and compare with the pattern and decide if it is part of the cultivar group or not, or if it is new diversity. And doing that, this work, we were able to modify, to correct about 20% of our data set where we had missing data, incorrect data set. We could also detect some aneuploids, so part of chromosomes that were missing in our sample. And we could also discover a new cluster of diversity. So this catalog is a result of a lot of curation with multiple scientists in the team. It's quite long, quite laborious. So we thought it would be good also to find a way to do that in a more effective or more efficient way. So our colleague Catherine with an intern started to develop a proof of concept based on machine learning, where we use those patterns that we convert into an image, a bitmap image, and then we use a deep learning model to make recognition of the pattern. So meaning that now we have like a classifier. So each time we have new accession that are genotype, we can go through this tool, and this tool is going to predict, to say, I think that this accession belongs to this cultivar group because of the curation we done before with the catalog. And if we don't have a good score, this is probably the priority accession that we are going to check to see if we don't have new diversity in our sampling, right? Okay. So this part was to show you how this method can be useful for Genebanks. But we believe that this type of work can be also useful for other type of application like direct use. Because I take the example of Pisangawak here. Pisangawak is a clonal, well, it's supposed to be a clonal group in bananas. It's an important cultivar. And we were quite surprised actually to detect multiple patterns. It means that we don't always have only one pattern. As a clonal group, we were expecting to have only one, but what we can see is that we can have different patterns that are resulting from different meiotic events. So some sexual event, are composing the group, and even different level of ploidies. So we have 3 patterns that are tetraploids, meaning that the parents were different. And that's become very interesting because in terms of morphology, they are very close. People recognize them as Pisangawak, but there is some diversity and some unsuspected diversity. And if you consider China, for instance, so it's a picture that one of our colleagues sent from Yunnan. Pisangawak is very important, very popular there. And they use probably only one clone, which is Fenjiao. So it will not be very difficult to introduce additional diversity of Pisangawak there because the plant will be quite similar, and to start assessing, what are the different traits that they are, if they bring more resilience to the field. So this is the type of thing that we could promote with a better understanding of the diversity inside the clonal groups. So we know that those Pisangawak are in China, but it's not always easy to know where are those cultivars. So we develop an online version of the catalog as well that we combine to a project that we have on citizen science. And basically, we retrieve a lot of pictures from multiple platforms, eNaturalist, PlantNet, Flickr, and so on. Some curation is being done by experts, mostly Christophe Génie working at IRAD. So we can really map these pictures to particular cultivar groups, and then we obtain some maps here. Those are the maps of the Pisangawak. So it's also a way to track where you can find the diversity on site and maybe target some intervention. So that's one of the strategies we could put in place with such a work. And one of the elements also I wanted to mention is with this mosaic work, you can look at the proximity between groups. And I take the example of the Cavendish here and the Gros Michel, which is also quite popular here in Colombia. And if you look closely at those patterns, those profiles, you should notice that there are 2 haplotypes that are exactly the same. So if you don't see it, I can understand it's difficult, but trust me. And it is a result because those 2 groups are actually half brother or half sister. They are very close. And we could see that we have another group, which is a diploid, Mshare, very popular in Tanzania, actually. And this Mshare group is actually one of the 2 parents that contributed to the Cavendish and the Gros Michel. And it is exactly the same mosaic, meaning that Mshare gave an unreduced gamete directly to create those groups. There was no recombination to do that. This is the type of observation we can make. And if we can do that for round races that were created long time ago, we can also use that for breeding. So it can be quite interesting to use it for breeding because breeding is very challenging in banana. There is a high level of sterility. It's difficult to get viable seeds. And when you do breeding in banana, you have a quite complicated scheme. So you need to start, for instance, our colleagues at IITA, they start with triploid bananas that they cross with diploid to get a tetraploid. Then they cross again a tetraploid with a diploid to get back to a triploid. So you have a lot of meiotic event with different level of ploidies. And it can take up to 15 years. So it's a long process, and it's very important to speed up this process in breeding for banana. And as a proof of concept, we took one accession in the Genebank, which is a breeding material, which is Goldfinger FIA01, which is a tetraploid. And we did the painting for this accession and as well for the parents, because we knew the pedigree. And looking at that, our colleagues in the team, Alberto Cianci, could compare those patterns, look at the differences, and was able to look at the recombination that occurred to be able to produce a gamete, the 1X gamete here, and did the same in the triploid banana to look at the second gamete. So with this system, we can track what happened during the meiotic recombination, and which can be quite useful.
Pues, buenos días, mi nombre es Santiago, tengo 20 años, me gradué del colegio ya hace 3 años, también tengo un...
Thank you. So I was thinking that it can be quite useful for breeding, because now you can imagine that you develop a breeding population. You can do this painting for the parents, you do the painting for the progenies, and you can look for specific events. So you can either look for a specific mosaic. You want to keep a very, very particular mosaic, like reconstructive breeding, or you may be interested by some integration and to look at very specific location on your genome, some QTL, and that you want to explore because it can bring some advantage to your crop, to your phenotypes. And doing that, you may also be interested to look at the genes that are behind that. So we can now look at the specific location on the genome. And the good news is that for all those, most of the wild species, the representative of the ancestral gene pools, we have now high quality genomes. We have good assemblies, we have gene annotation, we have transposable elements. This has been published recently. So we can go back to those ancestral genome and check if we don't have the full genome of the accession already available, because sequencing triploid is possible, but we don't have the genome of all triploid banana yet. I mean, it's still a challenge to produce. And all this information has been included in the Banana Genome Hub that we co-developed with CIRAD, where you can have access to a lot of tools to compare chromosomes together, to look at SNP marker, to look at genes, gene expression. So we have many different tools that are useful for gene discovery. And it has been useful to support the community for years. And it's even more useful for us now in the banana program because a few weeks ago, a new method for gene editing, which is tDNA-free in banana, was published. And we have now a very important tool, proof of concept, to make some edited lines in banana and to produce new lines, new edited lines with some additional traits. So I think that's an important breakthrough that maybe there will be a seminar on that later by Bart. It was developed by KU Leuven in collaboration with the Alliance and Bart Panis, the Genebank manager of ITC. And it's a very nice result, and it's a very exciting time to work in genomics in banana at this moment. So just to conclude, banana display a fascinating genetic diversity. We are convinced that there is much more to discover. There is still a lot of this diversity existing in Southeast Asia. And in silico chromosome painting was very effective for the curation and the classification of accession in Genebank. We believe that it can also enable some targeted use of this diversity directly, and also can boost molecular breeding strategies. So with that, I would like to thank in particular my team, Julie, Alberto, Max, Valentin, Catherine, that has been heavily involved in the work I have described before. The banana program with many names, Sébastien, Sijun, Nicolas, Bart, Ines, obviously Jorge, Lorena, that are here and organizing this Banana Day today. Our partners and all of you for a very warm welcome on the campus. Thank you very much. And happy to take any question.
Thank you. Thank you very much, Matthew, for such great presentation, especially in this day. I know that in Palmira campus, we will have more to learn about bananas during the morning. So it's good we're here. But for the people that are connected online, we are happy that you were able to join and learn more about this amazing work that is being done regarding bananas and everything related to this day also. So please, we open the floor for questions and answers, and I'm going to switch to Spanish for people that are connected. Well, you are already listening to interpretation, I'm sorry. If you would like to ask any question in Spanish, please do so. You can also write it down in the chat, and we can translate it for the English speakers. Thank you very much. You can also raise your hand and turn on your mic. Thank you, Marcela. Please go ahead. Marcela Quintero.
Yes, can you hear me?
Yes, very well.
Okay. Now, thank you, Sebastian and Matthew, for this great presentation, and happy Banana Day for everyone and for the Alliance. And I think all of us should be proud of the work that we do in banana, not only for being the custodians of the largest banana gene bank and then the diversity that is there, but also for keep advancing the science to be able to use that diversity. My question is regarding the chromosome painting methodology. And now, Mathieu, that you have been in the campus of Palmyra, working also with the team of Future Seeds, what opportunities you see in Future Seeds regarding applying similar approaches to the diversity that we are also safeguarding in Future Seeds? That's one thing. And the other thing is that that I found also very interesting is how all these advancements can also help to accelerate the breeding of banana. And I mean, how we can influence the banana breeding programs that are not in the alliance, are in IITA or in other organizations, how we can help them to accelerate their breeding efforts using all the advancements that you just showed to us. Over.
Okay. Thank you, Marcela, for these nice questions. So yeah, I have spent some time at Future Seeds. So we have discussed, and several members are here in the room, actually. So we have mostly discussed about data management for now, but we are using similar technologies with SNP. So there are options, obviously, for joint maybe methodology or analysis. But I think it's important first to understand exactly how the crop is being analyzed, like cassava and forages, and to see if the methodology can be applied in an effective way, because this methodology is well suited for crops that have like long generation time or not a lot of meiotic event, but it can be adapted to others. So I just prepared one slide in case of question about different crops, because just to show that it has been applied by other groups. The method is a little bit different, but it's the same principle. It has been applied in citrus, where they could also do the painting for the ancestral gene pool of citrus. So it makes also very nice drawing. And so there are trees, so long generation time. But it was also possible with another methodology to apply for rice. And in that case, we can also detect some introgressions that can be linked to some traits. And I think it can be applied to other crops. We could check for cassava, we could check for other crop, but it would require some investment in terms of time to really fine-tune the methodology. So there may be opportunity. Happy to discuss that in the future. And I think that's for the first question. For the second question, how to influence the breeding program? First, by doing promotion of what we do and what we can bring, and that's what we are doing. We have been approaching also IITA on this topic. I think they are interested by the methodology. The thing is that the institutional framework in the CGIAR, having Genebanks and breeding program not always funded by the same sources, doesn't help for this type of work that sits a bit in the middle, you know, these breeding activities. So no one knows exactly who should fund what, and it has been difficult really to find a project. But if we find a common framework to do that, I think we have a lot of opportunities to do that. And IITA published a publication recently where they were using part of the methodology with their population. So I think there is potential for that. I hope it replies your question, Marcela.
Yes, Mathieu. Yeah, that was very good. Thank you. Thank you. And also just would like to promote, I mean, how useful are these staff exchanges? I mean, for all of you, you may remember that last year we activated again in the Alliance staff exchanges to be able to, for example, in the case of Mathieu, to come to the campus in Colombia and start, you know, having an exchange with staff from Lever 6, from Future Seeds. So I think it has been a very good experience for Mathieu and also for the team here in the campus. So thank you also, Mathieu, for applying to the program, and I hope it has been also very beneficial for you.
Yeah, thank you very much for enabling this opportunity. I'm very happy to be here again. And I'm still here for about 1 month. So for people who want to talk to me on the campus, please take this opportunity. Yeah. Thank you very much.
Thank you, Marcela, and thank you, Mathieu. We have 2 questions in the chat. They were in Spanish. I'm going to say them in English. The first one says, very good presentation. Congratulations to the team. Do you have genetically edited Cavendish bananas with fertile male and female flowers aimed at more rapidly achieving hybrids crossed with diploids that are source of resistance to FOCTR4? I don't know. If so, how could we go about introducing these clones to Colombia? This question is from Romel. Romel, if you want, Romel from Agrosavia. And the question is in the chat also.
Yeah. Sorry, I will reply in English, but no, we don't. What I was referring to, and again, I'm not the expert on this, it was published a few weeks ago, is a proof of concept for the methodology for the gene editing, which is still DNA-free, but there is still some investment to make to develop additional edited lines that would be resistant to different traits like TR4. I don't know if Bart is online, actually, but he would probably be the best person to reply this question, or Sebastian. But at the moment, we don't have this to introduce in Colombia.
Okay. And he followed up that question, and he says, do you have any methodologies that are more feasible for evaluating ploidy, considering that equipment like flows, cytometers is expensive and scarce in Colombia? If so, who could I connect with the Alliance to begin this kind of work in the country? And Romel, you have your hand raised. Do you want to say something? Romel, puede activar el micrófono si desea.
Thank you very much. My question is about fertile male Cavendish, ¿ok?
Sí, puede decirlo en español.
Mi pregunta es si tienen editados genéticamente de Cavendish que pueden ser machos o que tengan polen fértil o hembras que también sean fértiles. Digamos que en Colombia estamos cruzando Cavendish con diploides con fuente de resistencia a la enfermedad, pero sé que ya han identificado algunos genes que se expresan en aquellos clones que pueden ser más fértiles. Entonces la pregunta era si tienen en algún banco de germoplasma algún clon tipo Cavendish que sea fértil tanto como macho o como hembra para seguir los cruzamientos con estos diploides que ya tenemos en el país que tienen fuente de resistencia a FOXR4T. Esa era la pregunta.
Jorge? No, entiendo, pero la pregunta es difícil. Pienso que no. No sé si alguna persona conoce pienso que no.
I think he's asking if in the Genebank
have
this.
Yeah, I don't think so.
Okay.
I don't think so. Or if it exists, it has to be evaluated. But resistant to TR4, no. But we probably need also more evaluation, but we don't have a specific clone that is known to be resistant to TR4 as Cavendish in the Genebank. I don't know why the camera actually, so I'm just moving my head like that, but
All the room has a camera. Sí.
Okay.
Romel.
Sí, no, es que, perdonen, es clones editados genéticamente tipo Cavendish, que sus madres, o sea, que tengan polen fértil o hembras fértiles, porque la naturaleza genética de estos materiales es que no producen semilla. Si hay algún editado genéticamente, pero para la parte sexual, más que la resistencia a FOXR4T. Esa es realmente la pregunta.
No sé. No sé.
No hay. No tenemos.
Okay,
We
gracias.
don't have those clones. We will translate the question said by Rommel and we will send it through the chat.
Yeah.
Thank you very much. Gracias, Rommel.
Monica?
Oh, okay. Hi, Mathieu. Thank you for the presentation. And it is really impressive to see all the different tools that you have developed for banana, for the characterization, and also how this can support conservation and probably the breeding programs. But I'm more interested now, and this is where my question goes, is about how all the data that you and the team has generated, the genomic information, how has this support a curation of the collection? So you mentioned this at the last part, and I think during your presentation you said that you identified some mislabeling around 20%. So I'm curious to see.
Not only mislabeling, but yeah.
Yeah, I'm curious to hear what were the actions taken after this identification, okay. And also if having all these tools and genetic resources, if you use this for genetic integrity checks due to the conservation. We know that in vitro conservation can cause changes in the genome. So if you use this for that. And the last one is, I know you have been in the conversation, but if you have assessed redundancy in the collection, if you know how similar are the materials, have different, now that you have this catalog, and if there have any actions taken to improve conservation and optimize resources.
Okay. For the curation, after this work was published a few months ago. So the first action was actually to develop a kind of dashboard where we compile passport data, field verification. So we send the plant to the field to get pictures that are reviewed by experts, this molecular information, ploidy information, and we combine this information to make some recommendation on curation. So what can happen is that there is mislabeling, so it's very easy to see that you have a pattern that is very different. Sometimes the information was missing, so it was important in the collection without this information, so we propose to add that this clone belongs to this cultivar group because it has exactly the same pattern. So we can formulate a number of recommendations to send back to genotyping if we suspect some problem between field verification and molecular characterization, because all those steps does not happen always at the same time as well. So you send a plant that was maybe extracted from the gene bank 10 years ago and one genotyping 2 years ago. So you have also to look at dates. But we have now, we have a kind of ecosystem with a dashboard where we can formulate some recommendation for action in the curation of the collection. And Ines, our curator of the collection, is looking at that to take some actions. So I think that's an important tool for that. For the aneuploidy, so what we do is that we check when we can where there is aneuploidy. And then it becomes a bit difficult to know if it is a result of an event in vitro or if it was the case before the introduction, like it exists in nature. So, but at least if we do that as soon as it is introduced, so we have a baseline, we can see if we have a loss of integrity or not at the beginning and monitor that maybe in 5 years, 10 years, and then we'll be able to take decision. So at the moment, except the plant was detected as totally off type, you know, the morphology is very strange. These one are removed, but otherwise we just note that there is aneuploidy. And for the redundancy, I think for banana it's quite difficult because you can have the plantain, for instance, that are very well known as well in Colombia, are very genetically similar, although they have a wide diversity morphologically because there is a lot of methylation behind that. So it's very difficult to take decision on redundancy based only on genetic information. It can be a tool to help, of course, but it's a whole process again. It's looking at passport data. Where does it come from? Maybe it was sent to a collection before and the collection sent to the, and by comparing all this information, maybe you can take some decision. But some discussion has been done, of course, in the gene bank. It's my colleague Julie who was being involved in that with Ines. And some action are being taken, but only genetic data are difficult. You can have a little change, you know, in the genetics and it makes a new trait. So the pattern that we see will not be sufficient to detect that change. So we see the big segment on the chromosome. So for redundancy, it's not ideal, I would say. Thank you.
We have a follow-up response from Sebastian in the chat. I'm just going to say it out loud for interpretation also. Flow cytometry is still the most efficient and cheapest method to determine ploidy level. As you have seen from the painting, also this advanced method determines the ploidy level even at LL resolution, but there are also morphological characteristics that are correlated to the ploidy level, such as size of stomata, stomatal density, growth, vigor, etc. Do you want any follow-up answer? Or okay. Perfect. Thank you, everyone. Thank you, Matthew. We have reached to the end of the seminar, but if you have any follow-up questions or comments, please send it through the chat, or after we will send an email with the digest of the seminar. We will also address these emails to the colleagues that are connected externally. So if you would like any follow-up comments or questions, we will make sure to send them to Matthew. And thank you very much, Matthew, for your time and for everyone participating today. Thank you.
Thank you. How many people were online?